Bismillahirrahmanirrahim. Now I am going to discuss about termination of transcription. I already discussed how transcription occurs. Now we know termination of transcription occur by row factor or without the row factor. Row factor has cutting activity. Suppose RNA polymerase, this RNA polymerase causes polymerization going forward and come to this side and stop polymerization. This row factor starts follow this RNA polymerase. RNA polymerase going forward, a row factor going following this RNA polymerase. Suppose RNA polymerase come to this point, and row factor causes cutting up to this point. RNA polymerase going forward, row factor going forward, and ultimately row factor come to this side, and RNA polymerase come to this side. Row factor also come to this side, and continuously following RNA polymerase enzyme and continuously cutting this strand. And we know this strand is RNA strand, and this nucleotide base also RNA nucleotide base, and this nucleotide strand is DNA, and this nucleotide base is DNA nucleotide base. So RNA polymerase causes polymerization continuously, and row factor by its cutting activity, it following this row factor follow the RNA polymerase and continuously cut this strand and separation of the RNA strand, and ultimately we get this RNA, and termination of the transcription occur by this row factor. This is called row dependent termination. Now what about row independent termination? Row independent termination is termination of the transcription process without the help of row factor. But how? You follow here this sequence, nucleotide base sequence: A, C, 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 G, 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 then C, 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 G, 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 U, U, U. This is called palindromic sequence. Palindromic sequence: A, C, 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 G, G, G. This is called palindromic sequence. You follow C, 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 G, 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 C, 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 G, G, G. So in between G, 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 this C, 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 in between G, 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 C, 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 there is attraction, force of attraction. There is C, 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 G, 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 there is attraction. So in between this nucleotide base, there is a attraction, and ultimately folding of this strand occur. When folding of this structure strand occur, we can get this structure, like this structure. Suppose here A also present, here U present. So we can get this palindromic sequence. This thing occurs in this strand in case of RNA independent termination. So when we get this sequence in this RNA, this sequence when we get in this RNA strand, this thing occur. This like a hairpin like structure develop in this strand. So RNA polymerase, suppose polymerase up to this area, hairpin like structure develop up to this area. RNA polymerase going forward, hairpin like structure developing and going forward also. But its folding structure carrying is not so easy by this RNA polymerase. That's why going forward up to a specific nucleotide base, RNA polymerase cannot go forward. RNA polymerase stop polymerization, and this palindromic sequence nucleotide base develop is like this hairpin like structure here. It causes separation as hairpin like structure backside there is suppose A or U or at forward U structure present. So adenine here is uracil. Suppose here U here A present. Ultimately there is adenine uracil double hydrogen bond. Here folding structure present, RNA polymerase stops here. So in between a U there is double hydrogen bond. It is weak bond and folding structure. Ultimately, this folding structure there is weak bond backside, some forward side. RNA polymerase cannot polymerize stop polymerization. So ultimately, this strand RNA strand becomes separated due to this folding structure becomes separated, and this we can get this RNA strand. This RNA strand helps in or used in protein synthesis. So it is called RNA independent termination of transcription. RNA independent, sorry, row independent termination, row independent termination of the transcription. So I discussed how row dependent and row independent termination occur. Row dependent termination, we know row factor has cutting activity. So RNA polymerase going forward by polymerization process and row factor following this RNA polymerase. RNA polymerase going forward, row factor continuously cutting this strand, a nucleotide base hydrogen bond, and strand is separated continuously. And up to this specific area, RNA polymerase stop polymerization, and row factor also cutting this strand, and this RNA strand is separated. This RNA strand we use to synthesize protein. How protein synthesize we will discuss in translation process inshallah. So up to this, how row factor causes transcription termination or termination of transcription process, and how without row factor termination of transcription is done, we already discussed. Now I am going to discuss about antibiotic rifampicin. How rifampicin causes in killing, how rifampicin can kill bacteria like Mycobacterium tuberculosis. We know Mycobacterium tuberculosis organism causes tuberculosis. Tuberculosis. So tuberculosis can be created by this rifampicin. We know RNA polymerase enzyme has beta prime, beta subunit, alpha alpha subunit, and also sigma factor. We already know that RNA polymerase enzyme enters into promoter region, Pibno box, or minus 35 hexameric nucleotide sequence. And when polymerization starts or transcription starts, sorry, not polymerization, transcription starts, this RNA polymerase enzyme become attached with this DNA nucleotide base. Beta subunit causes stabilization of the holoenzyme with the DNA strand nucleotide base, and beta prime causes making phosphodiester bond with the DNA strand of the holoenzyme. So if we inhibit this beta prime subunit by the action of rifampicin antibiotic, so phosphodiester bond between this holoenzyme and DNA strand is not possible, and polymerization is not possible. So RNA synthesis is inhibited, as well as ultimately protein synthesis is inhibited. This you follow here. This RNA synthesis process is inhibited in the Mycobacterium tuberculosis like organism. So ultimately, we can say that by rifampicin action, we inhibit the synthesis process of RNA, or transcription process is inhibited by rifampicin. So ultimately, RNA synthesis process is inhibited. RNA used in protein synthesis. Protein synthesis is not possible. So ultimately, bacteria is killed, or Mycobacterium tuberculosis is killed. We get rid of tuberculosis like disease. So by the action of rifampicin, we inhibit this beta prime subunit, and beta prime subunit helps in phosphodiester bond formation with this new coming nucleotide base, or this RNA polymerase enzyme. RNA polymerase enzyme. So this is the action of rifampicin. Thank you.